Hello class, we want to have our last lecture, we want to have the last section of our lecture on complements. So the complement system, this is what a series of proteins, also called complement components, that interact in a stepwise manner to destroy a pathogen. We have three ways. We have the what? Alternate pathway, classical pathway, then we have the lectin pathway. So let's look at it. So this is the classical pathway, and the classical pathway is mediated by what? Antibody binding to what? An antigen. And the antigen is what is located the particle. So the classical pathway is like this. We have anything you say classical means that is what came first. So this is the organism that we are seeing. This is its flagellum or flagella. Then we have the antigens here. So an antibody is this. It will come and bind. This is antibody. This antibody will come and bind to this antigen. It will also bind to another leg. This one leg. The second leg will also bind to this antigen. So this interaction between this antigen, the interaction between this antigen and this antibody, the bond between them will cause the complement proteins to be released to come and destroy this organism. So we have the classical pathway, this big polyvalent antibody you are seeing is immunoglobulin M. It contains 5 antibodies. Just one antibody, that's immunoglobulin M. M for mother, contains 5 antibodies, and it attaches to the antigens of this organism. So it leads to the production of C3, complement fragment C3. C3 will break down to produce C3b and C3a. C3b is what causes what? Coating. This is the organism. Then the C3b will come and coat its plasma membrane to make them attractive and recognizable to macrophage and neutrophils for them to be phagocytosed or destroyed. Then the C3a will go and mediate inflammatory reactions. Then we have the alternative pathway, that is the system, the complement proteins recognize the microorganism. Then the mannose binding lectin, this is lectin. Lectin is what is located, the blue structures on the organism. That is what we call lectin. So the organism will recognize this lectin. So when the organism recognizes this lectin, all these 3 steps will pass through. First, breaking down of C3 or converting C3 to C3b, which will go and coat the organism, some organisms, and destroy them, or C3a, that will go and mediate inflammatory responses or reactions. Then there will also be the production of C5, which will break down to C5b and C5a. For the C5b, we don't know its work as of now, but the C5a also goes to mediate allergic reactions. Then we have the membrane attack complex. The membrane attack complex, you can see the plasma membrane is here. but the complement will form a hole inside the plasma membrane of the cell. It's like this. There is a bacterial cell like this. This is a bacterial cell. This is the cell membrane. This is its cytoplasm of this bacterial cell. Then a complement protein will come and because there is complement, it will come and form a hollow tube inside this cell. So if you are an organism and you are there, if you catch a thief and you put hammer and nail the head of the thief will die. So when you the lectin pathway will lead to what? The membrane attack complex. That's the mark. Membrane attack complex. Membrane attack. Membrane attack complex. So the membrane attack complex will drill a hole. It will drill a hole inside this cell membrane of this bacteria. Bacteria or any organism at all. The complement system will drill a hole inside it and they can kill it. When they drill a hole inside you, that means that all the contents of the cell will leak out and the cell will die. So that is what we call membrane attack complex. So you can see this is the cell membrane. There's a membrane attack complex produced by these pathways, the mannose lectin binding pathway, that led to the total destruction. So now the steps for the destruction are: 1, C3A will mediate inflammation, that also fights organism. C5A, inflammation, that also kills organism. Because inflammation, there will be high temperature, and the organisms, they don't like high temperature. C3B will coat the cells of the organism so that they will be recognized by phagocytes and destroyed. Then the lectin pathway will lead to the membrane attack complex that will finally kill the organism. So let's look at it. I've explained these ones. So this is it. C6 to C9, that's the membrane attack complex. That will lead to cell lysis. Okay, so let's look at inflammation. When they say inflammation, okay, let's
Inflammation is the body's protective response against infections or injuries. Inflammation mobilizes defensive cells to the site of injury, limits the spread of pathogens, eliminates them, and initiates tissue repair. Inflammation can occur in any organ, but is most common and also most easily observable in the skin and underlying tissues. Typical signs include redness, heat, swelling, and pain. Inflammation is an important defense mechanism, but it can be a double-edged sword when things go wrong. An autoimmune disease may result when inflammation targets and destroys the body's own cells. An acute inflammation that fails to stop after the original insult is cleared can become chronic and damaging to healthy tissues. Acute inflammation is initiated when tissue-resident immune cells, such as macrophages, encounter an inflammatory stimulus. This stimulus can be a pathogen, a toxin, or an injured host cell. Binding of the stimulus to its receptor on the immune cell triggers a signaling cascade that activates production of cytokines and other inflammatory mediators. Inflammatory chemicals dilate blood vessels, increasing blood flow and enhancing vessel permeability, allowing plasma fluid and more immune cells to seep through and accumulate in the inflamed tissue. This vasodilation is responsible for clinical signs of inflammation such as redness, heat, and swelling. The infiltration of blood components into the injured tissue occurs in 3 phases. The first phase is the exudation of plasma fluid containing various antimicrobial mediators, platelets, and blood clotting factors. These factors can destroy microbes and stop any bleeding that may have occurred. The second phase is the infiltration of neutrophils, the major phagocytes involved in first-line defense. Once activated by inflammatory mediators, endothelial cells of blood vessels become adhesive. They attach to neutrophils in blood flow, slowing them down before getting them to squeeze through the vessel wall. Chemical cues guide neutrophils to the battlefield, where they engulf bacteria and destroy them with enzymes or toxic peroxides. Neutrophils may also release highly reactive oxygen species in a phenomenon known as oxidative burst, which kills pathogens faster and more efficiently. The pathogen-laden neutrophils then die via apoptosis. In the third phase arrive monocytes. Monocytes differentiate into macrophages, which then remove pathogens, injured cells, and dying neutrophils by phagocytosis. Macrophages that have completed their mission are cleared from the tissue by the lymphatic system. Accumulation of fluid increases pressure on lymphatic capillaries, forcing open their one-way valves, facilitating lymphatic drainage. Lymph-containing debris-laden macrophages passes through a number of lymph nodes and is filtered clean before it returns to the bloodstream. Once the site is cleared from the original insult, immune cells stop producing pro-inflammatory chemicals and instead start producing anti-inflammatory mediators, which actively drive the termination of inflammation. Many of these anti-inflammatory molecules are lipids, some of which are synthesized from dietary omega-3 fatty acids. This step is essential in ensuring the favorable outcome of inflammation. Failure to resolve inflammation leads to development of chronic inflammation, which continuously deals damage to healthy tissues. Chronic inflammation is a known contributing factor to pathogenesis of a wide variety of conditions, including cardiovascular diseases, asthma, diabetes, arthritis, and even cancer.
Of a group of serum consists of a complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properidin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Dear class, I'm very sorry, I was supposed to show the complement system and its pathway before showing the inflammation, because we've already finished tackling the complement system, but I believe the video I just showed is just going to be an enlightenment to augment the lecture I had on complements. But for now, let's look at the inflammation video. Let me now go and teach you something about inflammation. So let's go to inflammation slide. Okay, so all we saw is this, but this is inflammation. So one, when there's an injury, there's a splinter over here or a cut on the epidermis, you see. So bacteria will now invade when there's a tissue injury triggered by physical, chemical, or biological agents. This break in the skin caused by this splinter will now favor the transport of microorganisms into the skin, into the broken area of the skin, and enter the skin, and this is bacteria. So what happens? Step 2, there will be vasodilation. We have vasoconstriction and vasodilation. Vasodilation means that the blood vessels will dilate, will increase in thickness, in diameter. So if the blood vessel were like this, if this is the blood vessel is like this, it will now become dilated. So the diameter will increase to this. Diameter will now increase, the diameter will increase. So this is our new blood vessel. You see that this one, the thickness from here to here is bigger than from here to here. So this is our blood vessel. So if the diameter were to be 5 centimeters, this one will be about 7 centimeters. So there is vasodilation. Opposite of vasodilation is vasoconstriction. So this is opposite. So you can see this blood vessel is constricted, or vasoconstriction. Vaso means what? Blood vessel, or blood vessel, or constriction of blood vessel. So vasoconstriction, and this is vasodilation. Okay, so let's go back to our view. So here we have there is vasodilation, and when there is vasodilation, there is increased blood flow to injured area, provides increased delivery of plasma proteins, neutrophils, and phagocytes. So let's see. So when there is vasodilation here, yes, I'm tempted to ask you that blood flow through here and blood flow through here. Which one will have more blood flow? First one or second? I believe is the second one. Blood will flow more here because the blood vessel is dilated. You see the diameter is very big, 7, and this is 5. So more blood will flow here. Okay, so more blood is flowing in the second blood vessel because there should be what? It should provide plasma proteins, neutrophils, and phagocytes to fight the infection. So what happens is that there is also increased permeability. These are endothelial cells. These are cells that line blood vessels, the lumen of the blood vessel. So if this is the lumen of the blood vessel, this is the lumen. Lumen means the hole inside the blood vessel. It is the lumen. The cells that are lining this blood vessel are called endothelium. And most of them are simple squamous cells. So you can see blood vessel cell is here. So these are called endothelial cells. Endothelial cells. Endothelia. They are cells lining the blood vessel. Endothelial cells. So they are endothelial cells. Another one is here. So they are 2 endothelial cells. It's the same over here. So when there is inflammation, there's a break in this skin. Break due to a knife injury. It will introduce infection over here. So there will be what? It will introduce infection rather at the tissue. Tissues are not in the blood because it didn't go. The wound is not in the blood, it's in the tissue, it's in the epidermis. So in the epidermis, in the epidermal layer, so in the skin. So if this is the skin, overlying skin, this is the blood, this is the interstitial, that's outside the red blood cell, interstitial compartment. Interstitial compartment. So these are the bacteria. So when there is a break in this skin, these blood vessels now the cells over here will retract. There will be holes in there. There will be gaps between the cells. So the gaps will lead to the production of proteins, chemicals, cytokines to the site of infection over here. Then white blood cells or neutrophils in acute inflammation will travel and find a gap here, find a gap here, and enter it, and enter through the gap, to go and fight the infection. Another one will follow the same procedure, enter through the gap, and go and fight the infection. Another white blood cell, which is neutrophil, the same procedure, until the infection is destroyed by the cytokines produced by neutrophil or by engulfment or phagocytosis. So that is what we are seeing here. So we can see that the white blood cells are the yellow multilobed neutrophils. So they are moving. Here there will be protein-rich exudate containing immunoglobulins and complements. So immunoglobulin and complements will be released over here, and they are called exudates. Well, when plasma flows through blood vessel, the gaps in the blood vessel, and goes into the interstitial fluid section to go and fight an infection. And we call that, because it has proteins, that's immunoglobulins and complement inside it, we call it exudate. If it does not have proteins inside, we call it transudate. So here, if it does not have proteins inside it, we call it what? Transudate. Transudate. Okay, so let's continue. So our time is running out. So here, exudate proteins will follow to go and fight the infection. Then the white blood cell, that's the neutrophils, will also go and fight the infection. Then there will be phagocytosis over here. The white blood cells will phagocytose the bacteria, and the neutrophils and macrophages will also move to the side to in response to the injury. Okay, so the purpose of inflammation, what's the purpose of inflammation? Is to localize the infection. If there's inflammation, the infected area is localized so that the infection or the organism will not spread to other areas of the body. It will prevent spread of microbial invaders. It will neutralize toxins produced at the site. It will aid in the repair of damaged tissues. Okay, so the cardinal signs of inflammation are redness. Redness is called, when there is redness, meaning that, when we saw in the video, when there is vasodilation over here, you can see because more blood is flowing, that part of the tissue that there is inflammation or there is injury, that blood becomes very red. Imagine somebody, your husband slap you or hit your mouth. You can see that, or hit your face. You can see that all your face will be reddish. That part where he knocked will become very red, and it is inflamed because red blood cells will now flow there so that it will discharge neutrophils to go and fight the organisms that will prevail over there. Or wherever you have injury, the place will become very red. So apart from becoming red because of flow of blood to the injury site, it will also become very hot, and the hot is called calor. The redness is called rubor. The hotness is called calor. This is in Latin also. Then, because more blood will flow there, the blood will send some hot temperature there. And because of the trauma and the depolarization too, the place will be very hot. Then swelling, the place will swell, and the swelling over here is called tumor. Whenever you hear tumor, it is cancer. Cancer cells are called tumor cells. And the tumor, we have the benign tumor, the malignant tumor. We have benign. Benign means that it does not spread. It's not malignant. It does not spread. It is not a serious tumor or lesion. It doesn't spread. But the malignant tumor is what spreads. Malignant is what spreads. The tumor that spreads. And that one we call it what? Metastasis. We say it has metastasized. Metastasis. The tumor will dislodge from where it is and travel through the blood to other parts of the body. That's why if somebody gets cancer, we say that they will die in 3 months' time or 6 months' time, because the cancer cells are now floating all over the blood and spreading to other organs. But the tumor we are referring to, and the benign tumor, an example is what? Fibroid. It is localized. It does not spread. So we say it's fibroid. Fibroid is a benign tumor. But any cancer at all is a malignant tumor. But we are not referring to tumor cells. The tumor over here just means what? Swelling. Or edema. Edema is the same as what? Swelling. Other people spell it like this. Edema. Swelling. So we are not referring to cancer cells, whether benign or malignant. We are just referring to the place being swollen. So we have the swelling, that is tumor. Pain, that is dolor. Because there will be pain there because histamines will be prostaglandins will be produced. Prostaglandins And when you have headache or pain somewhere, it means that fatty acids have been broken down to produce what? Prostaglandins. Prostaglandins. So the prostaglandins cause the painful feeling we feel when we are injured, when you have headache. It means the body has produced a lot of prostaglandins and leukotrienes. Leukotrienes. That's they mediate pains. Okay, so we have pain, that is dolor. Functiolesa. Functiolesa means at the place that you are having the inflammation, there will be loss of function. So vasodilation will increase blood flow, that will cause redness or heat. Vasodilation will also will lead to stretch of endothelial lining and increased permeability. Leakage of plasma, swelling, edema. Vasoactive agents like prostaglandins and histamine will be produced, which cause pain. There will be nerve compression or nerve injury. Microglial toxins, irritation, pressure on the nerve, and by edema will cause pain at the inflammation side or the inflamed side. The reason why we have, when we say fever, it means that the body temperature is higher than 37 degrees C. That's the normal body temperature. But in feverish conditions, the temperature is more than 37. And the reason why we have fever is that it augments or the beneficial effect of inflammation. For when there is high temperature, your temperature is above 37, the microorganisms are supposed to be killed or inhibited by the high temperature. So God knows so best, He put fever in us, so that fever is the natural way the body responds to an infection by the hypothalamus increasing the temperature of the body, causing the person to feel feverish. So bad side effects of the fever will be malaise, body aches, and tiredness. So the rise in temperature will prevent the organism from staying. It will kill or inhibit the organism. But it's not, the high temperature is not also good for the enzymes in our body, because the enzymes in our body require optimum physiological temperature. So if the temperature of our bodies are so high, the enzymes are going to denature. So you see that COVID-19, somebody's temperature can rise to 42. The more the temperature rises, your enzymes will be dying. It will also affect your blood and your organs and other things. And you realize that you will pass out, you will die. So that means that there should be what, even though the temperature is high, due to the feverishness is high, but the hypothalamus, which is what, which is the restorer of the body temperature, is supposed to regularize or normalize the temperature. So we'll be looking at the hypothalamus in a nutshell. So you see, so the bad side effects, malaise, body aches, tiredness, is triggered by chemicals called pyrogens. So pyrogens are bacterial toxins, cytoplasmic contents of lice bacteria, antibody antigen complexes, and pyrogens. So these pyrogens, they trigger the feverishness. So let's see. So this is the hypothalamus. It is located just slightly above the midbrain, and is located under the thalamus. So that's hypothalamus. So the hypothalamus, this is section. When there is a wound over here, then information goes to the hypothalamus through the efferent artery, the efferent nerve. So the efferent nerve will deliver the information. This will lead to what? Resetting of the hypothalamic thermostat. It's going to control the temperature of the body. Nerve impulses cause shivering. So shivering is a metabolic process that will inhibit, produce sweating and vasoconstriction. That will inhibit bacterial growth. These processes increase body temperature to the point set by the hypothalamic thermostat. So in a nutshell, there should be what? The body temperature should be, when it increases due to inflammation, it should be restored to normal by the hypothalamus. Let's also look at acute phase proteins. Acute phase proteins. In my master's research, I worked on C-reactive protein. It was one of the markers I used, and it's an inflammatory protein. We also have proteins such as so acute phase proteins are produced in response to infection, inflammation, and tissue injury. They function by enhancing resistance to infection and promote repair of damaged tissue. I worked with C-reactive protein to determine inflammation in parasitic infection. There is also serum amyloid A deposition in kidneys, the liver, the heart. This also determines the extent of inflammation. There are protease inhibitors and coagulation proteins. Their levels in the blood will determine the extent of inflammation. So examples of the acute phase proteins are, we have the pentracins, and the pentracin C-reactive protein is part of it. We have collectins, and the collectins, the lectin component of the lectin mannose pathway of the complement system is over here. We have the ficolin, which acts as opsonin and activates the lectin pathway too. So lectin pathway is mediated by collectins and ficolins. Major functions of acute phase proteins are opsonization, complement agglutination, and complement fixation and agglutination of microbes, and extending range of farms. Let's look at cytokines in a nutshell. So cytokines are chemical mediators released from different kinds of cells, enabling cells to cell communication. And the cytokines include chemokines, interleukins, tumor necrosis factor, interferons. They are all cytokines. So in another lecture, we'll be looking at cytokines and other things. So in a summary, this is our summary. So fellow students, I would like to urge you to start after your Monday quiz, basic biochemistry practical quiz. Try and start learning the immunology on Monday, so that the things will not pile up, because the immunology is going to be very technical, even more than the biochemistry, the basic biochemistry practicals, the biological chemistry. The immunology is going to be very, very complicated. So start learning it. You can see that this lecture slide is loaded. Start learning it and understand it. The understanding is what I want, not you barbering it, by understanding the concept. When they say complement fixation, when they say opsonization, you have to understand it. When you hear inflammation, complement fixation, membrane attack complex, you should understand it. When you also hear phagocytosis, you should understand it. So go through the slides, watch the video over and over again. Know that you are here to study. You are here to study, because when you understand it, some of you are going to do your masters, some of you are going to do straightforward PhD from your first degree. You are going to the United States to do your PhD. Some will go to China, some will go to Canada, some will also go and do medicine. So please, some will also go and do masters in tech or even here. So please, whatever you want to do, get the foundation right, because biochemistry and immunology are the foundation courses for you to become a very good biomedical researcher. I would like to end today's lecture here. We will come back again with another lecture on adaptive immunity. Thank you very much for the audience you've granted me so far.